Good morning, my dear students. Please write your name and roll number in the chat box.
ठीक है sir.
Next. Last class we discussed about the protein drug delivery system, but how to formulation of protein, how to formulate the protein, and there is so many factors, especially the physical and the chemical. And the last class we discussed about the what are the physical factors to interfere with the formulation. And today we discuss about the chemical instability. What are the chemical instability? This chemical instability, that means there is a chemical problem that will interfere with the formulation of proteins. It involves the alteration in the molecular structure and producing a new chemical entity by formation or cleavage. Hello? हाँ बोलो. हाँ सब बुझते পারছি, বলো বলো, ক্লাসে আছি, বলো.
হ্যাঁ স্যার.
হ্যাঁ না 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 মনোজকে মনোজ 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 বলে তো আমি কাউকেই চিনি না. ও হো, সে আমাকে বলে করতে. না না সরি কিছু না, আমি বলো তোমার কোনো ইমার্জেন্সি হয়েছে না, তাড়াতাড়ি ধরে নিলাম. ভালো আছি তো, এমনিতে সব ভালো আছে, ঠিক আছে. তাহলে ওটা অন্য কারণ, আমি তো অনেক ফোন পেতে টাকা পয়সা করি, সেক্ষেত্রে হয়তো সে মানে একটা চিট করার জন্য, মানে ইচ্ছা করে আর কি, মানে সে দেখছে তো, বা তোমার সাথে আমার হয়তো রিলেশন আছে, মানে ফোনে সেই জন্য দেখছে. তোমার মানে ফোন পে আছে নাকি? হ্যাঁ ফোন পে আছে, ওই জন্য আমাদের এই কাজটা করছে. ঠিক আছে, ও পাঠিয়ে আনো. না না ঠিক করে নিয়েছো, মানে টাকা কি ওকে পাঠাতে বলছে? না 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 মনোজ বলে কাউকে আমি চিনি না, মানে এত অনেক স্টুডেন্ট আছে তো মনোজ নামে আমার কোনো. তুমি যদি পারো ওর নম্বরটা আমাকে শেয়ার, ওর নম্বর আছে যে? হ্যাঁ হ্যাঁ তোমার আমার তো হোয়াটসঅ্যাপ পাঠিয়ে দাও, আমি দেখবো কে দরকার ফ্রড হলে পুলিশে ইনফর্ম করবো. ঠিক আছে. Sorry to disturb. Hello. The involved in the alteration of the molecular structure and producing a new chemical entity by bond formation or by the cleavage. So suppose you prepare some formulation of protein, there is a chance of chemical modification of the structure and sometimes the chemical bonding or sometimes the cleavage of the proteins. So there is another instability problem in case of the protein formulation. And the stability of peptides and the proteins against the chemical reagents is decided by the temperature. That means the stability of the peptides, basically the most of the 90% above 90% protein which are formed by the peptide or peptide linkage, and this protein against the chemical reagent is decided the temperature, length of exposure, how much time of length exposure, and the amino acid composition, also their amino acid composition, even the sequence also, and the conformation of the peptide or the proteins. So these are the 3-4 factors which are stability of the protein. If any changes, either temperature, length of the exposure, amino acid composition, or the sequence of the amino acid composition, there is a chance of instability. And in that case, that stable formation can be unstable formulation, and there is no therapeutic activity will be given. So one of the great example, demediation. Demediation is the process by which the chemical instability or improper formulation will be formed due to demediation. What is demediation? Now this is the reaction involved in the hydrolysis of the side chain of amide chain. Demediation, that means amide chain will be removed. The side chain amide linkage of an amino acid residue leading to formation of free carboxylic acid. We know the amino acid contain amino group and carboxylic group. If the amide chain will be reduced, will be separated out, then only the carboxylic group is present. So this is called the deamidation, and in that case the therapeutic activity of protein, even the amino acid structure will be changed. So molecular structure change, their composition change, so their therapeutic activity will be changed. Another, asparginase and glutamate leading to conversion of neutral residue to a negatively charged residue and primary sequence isomerization. In case of asparginase glutamine, aspargine is coming from the due to deamination of aspartic acid, and glutamine, which is the deamination of glutamic acid, and leading to conversion of a neutral residue to negatively charged residue and primary sequence of isomerization. That will isomerize. So that will be, if the isomerization D to L or D to L, generally all of the amino acid L variety is the L configuration, or L variety is the or liporotatory is the optically active, or their therapeutic activity also. But if it is D, due to isomerization, there is a problem. Next, in vivo deamination, it is observed with the human growth hormones in vivo. Suppose these type of these two reactions which is occurred in the in vitro, but in vivo it is observed with the human cell growth, human growth hormone. Suppose growth hormone will be deamidated in that case, or bovine growth hormones, prolactin, adeno, corticotropic hormone, insulin, lysozyme and secretin. So these are the proteins, or mostly the hormones which are deaminated in vivo. There is a chance of instability. Ultimately the therapeutic or formulation will be unstable. Some of the factors which the main affecting the chemical instability. Number one, pH, because pH, all of the proteins of a certain or a specific pH. If the pH changes, then the activity or property, physical and chemical property will be lost. Another is the temperature. You know, if the above the temperature, the protein will be coagulated or denatured. So denatured or coagulated protein, or due to heat, protein activity will be lost. So in case of temperature high or low, there is a chance of modification, chemical modification. And another is the ionic strength, because all of a amino acid sequence of protein, they are having a positive charge or negative charge. Ultimately they were having a some isoelectric point. That is ionic strength, means there are isoelectric pH, isoelectric due to depend upon the ionic strength, because all these protein having positive charge as well as the negative charge, but after simultaneously they are having a some isoelectric pH, or at that pH they are the most stable condition. But if this pH changes in that ionic strength, there is a change of instability, or there is a problem of, in case of chemical bonding or chemical instability in case of protein formulation. The determination of a asparagine residue into the accelerate the neutral and alkaline pH. So now asparagine, which is residue to accelerated neutral and alkaline pH. Deamidation, neutral and alkaline pH. Basically it is amino acid, acidic amino acid. So it produces a neutral to alkaline, there is a problem. In case of tertiary structure of protein, also affect in the its stability, as observed with the trypsin, in which the tertiary structure prevent the deamidation. So deamidation is a factor basically done by the pH, temperature, ionic strength, and in the deamination of the acidity. So these are the factors which can aggravate the deamination, and if deamidation, if the deamidation is occurred, there is a formulation will be unstable. Next, the method to prevent the deamination. How to prevent? If the substances or the protein prone to deaminated and how to prevent it. The use of genetic engineering methods or by recombinant DNA technology. There is a two way, either genetic engineering, you can prepare it, the protein, or by recombinant DNA technology. The two technology you can use. Only the simple technology you can just prepare the protein isolation and purification, after then it will be used. That is not for preventing this deamination, but in case of genetic engineering and recombinant DNA technology, you can use this. There is a less chance of deamidation or no chance of deamidation. The asparagine residue can be selectively eliminated and replaced by the others residue and provide the conformation, and the bioactivity of protein can be maintained. So if you can eliminate it, the deamination, the bioactivity of the protein can be maintained. Another, number 2, chemical instability due to oxidation and the reduction. And I think this is the most commonest. Oxidation is the most common occurred during the isolation, synthesis or storage of protein. In case of oxidation, oxidation means oxygen incorporated into the protein. But this oxidation is common, occurs during the isolation, when the protein can be isolated, that means separate, or maybe synthesis, or maybe storage of the protein, and the oxidation will be done. So if the oxidized protein, there is the same problem will be done. But what factors fit aggravate the oxidation? The oxidative degradation of reaction can even occurs in the atmospheric oxygen under the mild condition. The oxidative degradation reaction can even occur in atmospheric oxygen under the mild condition, or it is called the auto-oxidation. Suppose the protein which is just kept into the air, the oxygen will come and it will be oxidized. That is called the auto-oxidation. And the temperature, sometimes the temperature which aggravate the oxidation procedure, and the pH also same, and the traces amount of the metal ion and the buffer influence this reaction, and that means oxidation. The oxidation takes place involve the side chain of histidine, lysine, tryptophan, tyrosine residue in the protein. So in case of oxidation, especially it will be occurred or chances in case of histidine. You suppose you are prepared some histidine which are used as some therapeutic effect or used as a drug, but there is a chance of oxidation in case of histidine like substance or lysine or tryptophan or tyrosine residue. Basically tryptophan, tyrosine are the aromatic amino acid, and the histidine, lysine are the basic amino acid. That means basic and aromatic. These are two types of amino acid which are prone to chance of oxidation. Number 5 is the groups, the methionine, thio groups, actually thio S group of methionine is particularly susceptible to oxidation. When a methionine is susceptible to oxidation under the acidic condition, methionine residue can oxidize by the atmospheric oxygen. So in that case, under the acidic condition, not in the basic condition. In acidic condition, there is a chance of oxidation of methionine oxidized by the atmospheric oxygen. So methionine, if is suppose a protein containing methionine, so there is a chance of, if it is kept in the air or in presence of air or in presence of the oxygen, there is a chance of oxidation. So that would be oxidation is the most common factors or chance of oxidation, and that will be damage the drugs. I think, do you understand? If not, please ask me the question. Go through the next slide. Any question up to this amidation, oxidation? Hello? Any question up to this 2 slides? Anybody can tell? Do you understand?
Yes, sir.
Do you understand up to this oxidation and amidation? and how to prevent the amidation and how to prevent the oxidation. Hello?
Yes, sir.
Do you understand?
Yes, sir.
Okay. Another is the oxidizing agent, like sometimes use some agents with the protein, some hydrogen peroxide, dimethyl sulfoxide, iodine can oxidize the methionine to methionine sulfoxide. So suppose some oxidizing agent in presence of the protein, there is a chance of oxidation. The thiol groups of the cysteine can oxidize to sulfonic acid, and oxidation by the iodine or by hydrogen peroxide is catalyzed by the metal ion and may occur in the spontaneously by the atmospheric oxygen. So that can be this hydrogen peroxide, iodine or oxidizing agent may be prevented away from the proteins. Usually the oxidation of amino acid residue is followed by the significant loss of biological activity as observed after oxidation of met residue in calcitonin, corticotrophin and gastrin. And glucagon is the exception as it retains the biological activity even after oxidation. This is the another good stability in case of glucagon, because after it is oxidation there is a no loss of property, but other things, other protein which are lost their property if they oxidize. Method to prevention of oxidation and reduction. So obviously antioxidant, the oxidation scavengers, you don't know, suppose some in case of some oils, hair oils, or you use this, there is some use some antioxidant to prevent the oxidation. Here in case of protein formulation, if you use some antioxidant, the oxidation scavengers, that means to oxygen will be removed or maybe block this action, acid or base catalyzed by the oxidation. For example, phenolic compound, propyl gallate. Phenolic compound and the propyl gallate, which are the compound which prevent the oxidation or which are used as a oxidation scavengers. Another reducing agent, sometimes there is a reducing agent, that means to prevent the reduction. One the ascorbic acid, sodium sulfate, thioglycerol and thioglycolic acid. Ascorbic acid, sodium sulfate, thioglycerol and thioglycolic acid. These are the four agents which are used as a reducing agent to prevent the oxidation, and some chelating agent also which inhibit the oxidation reduction. Number 1 EDTA, ethylene diamine tetraacetic acid, citric acid, nitrogen plus, refrigeration, protection from the light and adjustment pH. So these are the factors you can control it. There is a less chance of oxidation and reduction. And avoiding the vigorous stirring and inclusion of air by dispersing the solvent can prevent the air intended the oxidation. So these are the method you can apply it when suitable protein formulation, and the stable protein formulation can be achieved. Otherwise it will be a problem. Do you understand?
Yes, sir.
Okay. Next is the proteolysis. First is the amidation, then oxidation, reduction. Fourth is the proteolysis. That means breakdown of or hydrolysis of protein peptide bonds. The hydrolysis of peptide bond within the polypeptides or protein destroys, at least reduce in the activity. So proteolysis can be prevented, otherwise the protein activity will be lost. And what are the factors which favor the proteolysis and prevention? Some factors which aggravate the proteolysis and some factors which de-aggravate or prevent the proteolysis. Number 1, exposing the proteins to what's the condition and the extreme of the pH or high temperature or the proteolytic enzyme. Obviously the proteolytic enzymes can breakdown of the peptides or proteolysis will be done, or the extreme pH, either acidic, either basic, or in the high temperature, that means proteolysis will be break down the peptide linkage. Another chance of bacterial contamination. If the bacteria contaminated with the protein, there is a chance of proteolysis, because the bacterial proteolytic enzyme which degraded the or hydrolyzes the peptide linkage. Another is the protease enzymes may be gain excess during the isolation, purification, recovery of recombinant protein from the cell extract or culture fluid. In case of protease, protease is the enzymes which also gain access during the isolation, purification, recovery of recombinant protein. Suppose you are prepared in the recombinant genetic proteins, and their isolation, purification and recovery in presence of protease from the cell extract or the cell culture fluid, there is a chance of proteolysis, and this problem can be minimized. So there are the three main causes, the proteolysis, but it can be minimized it by the manipulation of the solution condition during the stage of purification, or addition of the protease inhibitors. In case of purification or addition of the protease inhibitors, you can use it. And some proteins have in the autoproteolytic activity, and this property aids in controlling the level and function of protein in vivo. Autoproteolytic activity. Number 5 is the disulfide exchange. Some protein, all protein, most of the protein is the peptide linkage, but others protein sometimes disulfide SS bond. These thiols are disulfide exchange, showing the linear intermediate in which the change in the shear amount the 3 sulfur atoms. And the thiosulfate, the thiolate group attack a sulfur atom of disulfide bonds and displacing the other sulfur atoms and forming a new disulfide bonds, and this results in a alteration in the three-dimensional structure and followed by a result exchange in the biological activity. And this reaction occurs in the neutral or alkaline medium. And accelerates by the thiols, and which are generated by the disulfide exchange. Method of prevention of disulfide exchange. How to prevent the disulfide exchange? By thiol scavenger. Even in case of oxidation scavenger, here is the thiol scavenger, such as para mercury mercury benzoate or copper ions. Paramercury benzoate, which is thiol scavenger. Antioxidant, which is the oxidation scavengers. Here the oxygen scavengers. Thiol scavenger, be careful, remember this name. What is thiol scavenger? The paramercury benzoate and copper ion, which prevent the sulfide exchange. Next is the racemization. I think up to this clear it. If any problem, you can ask me. You stop me and ask me the question. If any sort of problem. Up to this clear? Hello?
Yes, sir. Yes, sir, clear.
You understand? বুঝতে পারছো? Some problems, chance of degradation of protein. But if it is degraded, there is a therapeutic activity of loss. So which factors or which process to degrade the protein, and how to minimize the problem? Okay. Last class we discussed about the physical instability, and today is discuss about the chemical instability. What are the chemical regimes to destroy the protein? Next is the racemization. Racemization, what is? D to L or L to D. It is the alteration of L amino acid is converted to D, or D amino acid or L mixture, with the exception of glycine in the mammalian Only the exception of glycine. Why glycine? But DL isomerization occurred. This is the problem. Generally, all LIVO isomer is the optically active, but dextro isomer is totally optically inactive. Optically inactive, that means therapeutically inactive, and their activity also lost. But exception is the glycine. Why? In case of glycine, racemization is not possible. কে বলতে পারে? Only the exception. Out of 20 amino acid, only glycine is the exceptional case. Otherwise, there is a chance of all 19 amino acid, there is a chance of racemization. And why? Hello? Why glycine is not? কেন গ্লাইসিনের ক্ষেত্রে রেসিমাইজেশন হচ্ছে না কেন? কে বলতে পারে? এনিবডি ক্যান টেল? অভিক, উজ্জ্বল কে বলো কেন গ্লাইসিনের ক্ষেত্রে রেসিমাইজেশন হওয়ার চান্স নেই? অরুণাভ, জিজ্ঞাসা করছি তখন লেফট করে যায়। বলো গ্লাইসিনের ক্ষেত্রে রেসিমাইজেশন হওয়ার চান্স নেই, অন্য অ্যামাইনো অ্যাসিডের ক্ষেত্রে চান্স রয়েছে। হয়
স্যার
কি মনে হয় বলো?
একটাই আছে, রেসিমার আছে স্যার, এভিমার আছে স্যার। একটা গ্লাইসিন একটাই অপটিক্যাল আইসোমার আছে না?
Glycine actually optically inactive.
হ্যাঁ স্যার, মানে
The glycine is optically inactive. মানে কোনো না D হয় না L হয়, because they are having a no asymmetric carbon atom. কারণ একটা আছে CaHH, আর another হচ্ছে NH2, and দুটো হচ্ছে হাইড্রোজেন। এটা হচ্ছে সবচেয়ে সিম্পলেস্ট অ্যামাইনো অ্যাসিড হচ্ছে গ্লাইসিন। and যেহেতু চারটে বন্ডের ডিফারেন্ট হলে তবেই ইট ইজ অ্যাসিমেট্রিক কার্বন অ্যাটম। If they asymmetric carbon atom, there is a chance of optical isomer, DL isomer. But if the glycine having a no asymmetric carbon atom, so there is a chance of optical activity. So ultimately, it is a optically inactive. So optically inactive substances or isomerization DL এর মানে তার না D আছে না L আছে, তো আর DL এর কনভার্সেশন হওয়ার কোনো চান্সই নেই। ক্লিয়ার?
হ্যাঁ স্যার।
Actually, গ্লাইসিন অপটিক্যালি ইনঅ্যাক্টিভ, আর গুলো সবাই অপটিক্যালি অ্যাক্টিভ। মানে ওদের এল ভ্যারাইটি ফিজো, হ্যাঁ মানে বাকি 19 অ্যামাইনো অ্যাসিড হচ্ছে অপটিক্যালি অ্যাক্টিভ, বিকজ দে হ্যাভিং এ অ্যাসিমেট্রিক at least one asymmetric carbon atom. And there is a chance of one D, one L variety. L variety is physiologically active, but D variety physically inactive. But when the L is converted D or D is converted to L, there is a chance of isomerization. If isomerization occurred, then the therapeutic activity or biological activity will be lost. But in case of glycine, it is optically inactive. That's why there is no isomerization. তার অপটিক্যাল রোটেশনই যদি না থাকে তাহলে আর আইসোমারাইজেশন মানে ডিএল এর চেঞ্জ হওয়ার চান্স তো নেই। Am I clear?
হ্যাঁ স্যার।
হ্যালো,
হ্যাঁ স্যার,
ক্লিয়ার। ঠিক আছে, আরে না বুঝতে পারলে বলবে তো।
হ্যাঁ স্যার, বুঝতে পেরেছি।
হ্যাঁ হ্যাঁ বল। Next, racemization may be peptide bond that are sensitive to proteolytics enzymes. This racemization, in case of proteolytic enzyme, they are very much sensitive because there is a chance of D to L or L to D, vice versa. And this reaction can be catalyzed in the neutral or alkaline media by thiol, and which may be arises as a result of hydrolytic cleavage of disulfide. So racemization is the one of the, তোমরা শুধু ফ্যাক্টর গুলো মনে রাখো and how to minimize the factors in case of, এটা ক্ষেত্রে কোনো পার্টিকুলার ড্রাগের ক্ষেত্রে আমি ইন জেনারেল I discuss with you. Suppose you prepare a one formulation particular protein, in that case which chemical factors influence or changes the drugs, that will be you can only use. সবগুলোই চেঞ্জ করতে হবে বা করতে হবে তার কোনো গ্যারান্টি নেই, বাট দিস ইজ দ্য জেনারেল pathway by which the protein will be degraded by unstable due to some chemical reaction. And how to prevent method of preservation of racemization, মানে how to prevent the racemization can be prevented. The thiolated ions carry out the neutrophilic attack on the sulfur atoms of the disulfide linkage. An addition of thiol scavengers, same compound, be careful this, remember this compound, P মানে para mercury benzoate, para mercury benzoate, and the N thilamidine and copper ion. Basically these three ions which are prevent the susceptible to sulfate or sulfide. Okay, then is the beta elimination. The mechanism in which the beta elimination is similar to racemization, that is it produced through the carbanium intermediates. At the higher the elimination rate reveals under alkaline condition, which ultimately lead to the loss of biological activity. So the beta elimination, actually beta plated seed or beta chain, in that case the beta elimination is similar like a racemization, and if the beta elimination is occurred, there is a chance of biological activity, loss of biological activity. And protein residue susceptible to beta elimination under alkaline condition like cysteine, lysine, and the serine. So you can remember it to prevent the beta elimination. So commonly the use excipient, sometimes not only these are the physical and the chemical parameter which affect the stability of protein formulation, but sometimes there is some excipient, that means additives, which are produce some instability for solving the physical and chemical stability problems. Commonly use excipient. Some here also this excipient used to minimize the problem. Number one, surfactant function to prevent the degradation or aggregation, denaturation and aggregation. Now surfactant, because surfactant which decrease the surface tension and prevent the denaturation and aggregation, because there are two methods which are very much unstable of the protein. For example, polysorbate 20 and 80. In case of, do you know the polysorbate 80 and 20? Hello?
হ্যাঁ স্যার,
Which agent? Polysorbate 20 and 80? Which type of agent is this? Obviously the class of surfactant, but especially which class? কিসের জন্য ইউজ করা হয়? In case of pharmaceutical formulation, polysorbate 20 আছে, 40 আছে, 60 আছে, 80 আছে, সেরকম সরবিটান এস্টার 20, 40 and 60 and 80, মানে অ্যাকচুয়ালি 20 to 80 ইউজ করলে এই চান্স গুলো থাকে না। নেক্সট কি বলতো, এটা কি কাজে ইউজ করে পলিসার্বেট 80 and 20? ইট ইজ ইউজড এজ ইমালসিফাইং এজেন্ট।
হ্যাঁ স্যার, আপনি
স্টেবিলিটিতে স্যার ইউজ করা হয় তো পলিসরবেট,
হ্যাঁ স্টেবল করার জন্য।
সার্ফেক্ট্যান্ট না স্যার, ও আচ্ছা ইমালসিফাইং এজেন্ট তো। হ্যাঁ
অ্যাকচুয়ালি এটা কম্পাউন্ডটা কি ইমালসিফাইং এজেন্ট ফর স্টেবল ইমালশন তৈরি করার জন্য, যেহেতু অয়েল ফেজ আর ওয়াটার ফেজটা যাতে সেপারেট না হয়ে যায়, সেই জন্য আমরা সার্ফেক্টেন্ট ইউজ করি।
স্যার 20-80 টা কি ওই স্টেন তো? ওটা স্টেন তো
20-80 এটা হচ্ছে না স্ট্রেংথ না, এটা হচ্ছে বিভিন্ন অলিগ হচ্ছে 20 হচ্ছে
পলিসরবেটের স্টেন স্যার।
হ্যাঁ এটা হচ্ছে ফ্যাটি অ্যাসিড, মানে কোন ফ্যাটি অ্যাসিড থাকে, এটা হচ্ছে 20 টা হচ্ছে অলিয়েট, তারপরে পামিটেট, তারপরে মানে 60 টা হচ্ছে স্টিয়ারেট, 80 টা হচ্ছে স্টিয়ারেট, মানে কোন ফ্যাটি অ্যাসিড, মানে ওই পলিঅক্সি ইথিলিন অফ অলিয়েট, স্টিয়ারেট, পামিটেট। ওকে, এর উপরে ডিপেন্ড করে এটা সরবিটান এস্টার অফ ফ্যাটি অ্যাসিড স্প্যান 20 আছে, 40 আছে, 60 আছে, এটা পলিঅক্সি ইথিলিন, আর হচ্ছে সরবিটান এস্টার অফ ফ্যাটি অ্যাসিড, মানে সরবিটান এস্টার অফ অলিয়েট, পামিটেট, স্টিয়ারেট। ঠিক আছে, এর উপরে ডিপেন্ড করে এই 40-60 ডিপেন্ড করে, কিন্তু এর কিন্তু ওটা বা মলিকুলার রেট বা অন্য কিছু নয়, কিন্তু এটা তো জানার দরকার ছিল। এটা হচ্ছে, এখন বুঝতে পেরেছো? হ্যালো, ইট ইজ ডিপেন্ড আপন দা ফ্যাটি অ্যাসিড। ওই 20 এর মধ্যে থাকে অলিয়েট, একটা হচ্ছে স্প্যান এন্ড টুইন, তোমরা খুব কমন নামে পরিচিত। পলিসরবেটটা হচ্ছে টুইন, TWEEN, টুইন।
হ্যাঁ 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 স্যার।
স্প্যান এন্ড টুইন মানে খুব কমন নামে যেটা পরিচিত সেটা হচ্ছে স্প্যান এন্ড টুইন। এই টুইনটা হচ্ছে পলিসরবেট, পলিঅক্সি ইথিলিন অফ অলিয়েট, আর এটা হচ্ছে সরবেটান এস্টার অফ অলিয়েট। সেটা হচ্ছে 20 অলিয়েট, পামিটেড 640, স্টিয়ারেট 60, 80টা অ্যারাচিডোনেট সম্ভবত। নেক্সট হচ্ছে এনাদের হচ্ছে অ্যালবুমিন। অ্যালবুমিন কি করে? টু অ্যান্টি এগ্রিগেটরি এজেন্ট, মানে টু প্রিভেন্ট দ্য এগ্রিগেশন অফ প্রোটিন in case of serum albumin. Example, serum albumin. Next, sugars. Some sugars which also prevent this chemical reaction and stabilize the proteins against the denaturation. Say for example, mannitol, polyene glycol, sucrose and lactose. Another, cryoprotectants. Cryoprotectants means stabilize the protein against the very cold condition. In case of high temperature, there is a degradation of protein. In case of cold temperature, there is a degradation of protein. So in that case, how to prevent? The stabilize the protein against the very cold condition. Say for example, sugar. Example material is the sugar, amino acid, amine, polyols, and the salt. Another, like the same substances which are used to prevent the chemical instability, osmolarity agent, stabilize pH. Example, phosphate, citrate buffers. Number, preservatives, prevent the microbes. That means if the microbial attack of protein, there is a chance of degradation of protein. And in that case, phenol and benzyl alcohol is used. Phenol and benzyl alcohol is used. Next is the antioxidant, is the very common compounds. Prevent the oxidation. Number 1, ascorbic acid, sulfide, cysteine, and chelating agent to prevent the oxidation. Next, solubility enhancing agent. Obviously, those compound formulated that must be soluble with the water. Otherwise, it will be not absorbed, not distributed, not bioavailability, not the therapeutic activity. To increase the solubility of the protein, as for example, amino acid, detergent, and cyclodextrin. Cyclodextrin, 2 compound which are used for solubility enhancing agent. Next is a lyoprotectant. That means stabilize the protein during the freeze drying. ওটা ছিল during the cold temperature, and lyoprotect সেটা হচ্ছে cryo, এটা হচ্ছে lyoprotectant stabilize the protein during the freeze drying. In case of freeze drying, there is a chance of degradation of protein that will be prevented by the lyoprotectant. An example is very common. We remember if the multiple choice question come this chart থেকে, এই chart থেকে, from this chart I give a question. The sugars is used as a lyoprotectant. Next, parental route of administration. Parental route is the most effective way to systemic delivery of proteins and the peptides. Because why parental route is choose? Because in case of the protein is administered orally, there is a chance of degradation. Because when you get to the stomach, the proteolytic enzyme pepsin, which is present in the stomach, and that will be degraded the protein. That's why most of the protein preparation which are in the parenteral form, and this is best choice to achieve the therapeutic activity. And mainly 3 routes of administration. One is the intramuscular route, I think so all of you know. Intramuscular, intravascular, blood vessels and muscle, তার মানে IV, and subcutaneous. Intravascular means vascular মানে IV, IV, intramuscular or subcutaneous. These are the 3 preferable route of administration by which you can administer the protein formulation. What are the advantage or disadvantage of this route? Number 1, routes of delivery of 95% to 99% of the protein drug will be delivered. Number 2, allow the rapid and complete absorption, because in case of parental preparation, the rate of absorption is 100%. So their viability also 100%. Another is the most important, to avoid the fast pass metabolism. That means they are not goes to the liver, and the liver is metabolized. So fast pass metabolism can be prevented by this route of administration. Do you understand what are the advantage of these three routes? Fast pass metabolism, do you know? Hello?
হ্যাঁ স্যার,
What is fast pass metabolism?
স্যার লিভারে
কি বলো তো?
হচ্ছে, লিভারে
হ্যাঁ।
স্যার, লিভারটা
তা
স্যার
ফাস্ট পাস বলছে কেন? ফাস্ট কেন বলা হচ্ছে? বিকজ মোস্ট অফ দ্য ড্রাগ ইফ ইউ টেক orally, it will goes to the liver and the metabolism start. That's why it is the fast metabolism. আর যেহেতু এটা ফাস্ট পাস, মানে ফাস্ট পাস মেটাবলিজম অ্যাভয়েড করছে, কারণ ডাইরেক্ট ব্লাডের মধ্যে দিয়ে দেওয়া হচ্ছে, দ্যাটস ওয়াই ইট ইজ কল্ড দ্য ফাস্ট, অ্যাভয়েড দ্য ফাস্ট পাস মেটাবলিজম। না, জেনারেলি ফাস্ট মেটাবলিজম ডান বাই দ্য লিভার if it is administered through orally. So generally the protein which are administered into the parenteral route, so there is a fast pass metabolism will be reduced, but no fast pass metabolism. Clear? তার মানে এই সেই জন্য এই ক্ষেত্রে তিনটে রুট বেশি প্রেফারেবল। এবং এখনো পর্যন্ত যত প্রোটিন ড্রাগ ডেলিভারি সিস্টেম all are in the parenteral, exception in the only the oral OPV, oral polio vaccine. Another disadvantage, obviously, প্রত্যেকের কিছু অ্যাডভান্টেজ থাকলে obviously there is some disadvantage. The problem with the overdosing or necrosis. Suppose there are 1 ml, 2 ml, 100 mg, 1 gram, that will be better. But a continuous process, suppose given some protein, in that case there is a problem of overdosing. Number 2, the local tissue reaction or hypersensitivity. Sometimes the local tissue reacted, when in that case where it will be injected, this local tissue will be damaged sometimes, or if you continuous use. Another is the hypersensitivity, because the protein is an antigen or any foreign protein which are administered to the body, there is a chance of hypersensitivity reaction, say delayed or a immediate hypersensitivity reaction will be a chance. Everyone hates get a needle. Everyone hates getting a needle. Actually কেউ ইনজেকশন নিতে ভালোবাসে না, এটা সত্যি কথা। যদি সেটা ট্যাবলেট হয়, সবাই অনেক প্রেফারেবলি অনেক বেশি। একই মেডিসিন যদি ট্যাবলেট আকারে পাওয়া যায়, আনকনসিয়াস না হয়, তাহলে কিন্তু সে ইনজেকশন নিতে চাইবে না, ট্যাবলেটই নিতে চাইবে। এটাই তো কমন কনসেপ্ট বা কমন মানুষের কি বলে ইচ্ছে, আমরা তো সেটাই থাকে না আমাদের মধ্যে। যদি ট্যাবলেট পাওয়া যায়, প্যারাসিটামল ট্যাবলেট যদি পাওয়া যায়, শুধু ট্যাবলেটই ইচ্ছে বেশি লাগে, তাই না? দুটো ফর্মে পাওয়া যায়, সেক্ষেত্রে আমরা কেউ কি ইনজেকশন নিতে পছন্দ করি? কেউ করবো না। কমন কি অ্যাকসেপ্টেন্স সবসময় ট্যাবলেট, ক্যাপসুল, সিরাপ এইসবই বেটার। নেক্সট ধরো ইন্ট্রাভেনাস রুট, excessively metabolized and the tissue drug bound into the site of IM intramuscular can be administered by this route, say for example insulin or interferon. Disadvantage, cause pain, tissue necrosis and thrombophobia, and advantage is the in case of antibiotics can be administered by the intravenous route. Then intramuscular gamma globulin, which is given in this route, gamma globulin, and proved to that the long-term protection from the hepatic infection. And some drugs grips the roots, including the long-acting insulin and growth hormone. And what are the disadvantage? Not used for all proteins, peptides drug because of metabolism of drugs at the site of injection. Subcutaneous route, control release is obtained from the implantments, polymeric devices, and these are the preferred for the cross-linked polymers which are biocompatible or biodegradable. Polylactic acid, and release of insulin, bovine serum albumin, LH were prolonged by this route of administration. Next, the parenteral drug delivery system, the polymer based on the polymer-based drug delivery system. So many discuss elaborately polymer-based only protein with polymers, liposomal-based drug delivery system, hydrogel-based drug delivery system, emulsion-based drug delivery system, and generally different pumps like implantable infusion pumps and mechanical pumps are used for this preparation. The preparation for the route of proteins and peptide drug can be administration by these dosage pumps like liposomes, aqueous drug lipid layers, like hydrogel, drug enclosed into the gelatinous polymer, emulsion, hydrophilic lipophilic drugs, cellular circulation, enclosed in the erythrocytes, and the self-regulated devices like competitive desorption, enzymatic substrate reaction, eruption control, and the temperature sensitive drug. In that case, the self-regulated devices are used for these 4 types of preparation. And in case of pump, the implantants, mechanical and control release dosage pump by this pump drug delivery systems. Number 1, the polymer-based drug delivery system. The polymer are used to carrier in the drug delivery system. Generally, in case of pharmaceutics, you can use hydrogel 60-40 different types of polymer you can use for the formulation of the different types of drugs. So here only drug which is in the protein in natures. So polymer is used for this, used as a carrier or to prevent the degradation of the drugs. Characteristic of the polymer, it must be biodegradable because it will be produced into the body. It should be biocompatible if the polymer do not hamper or any react with the body, and non-toxic, non-irritating, non-sensitizing. There is no reaction, non-toxic, no types of reaction, any sort of reaction cannot be done by the polymers. That type of polymer you can choose and formulate it into the proteins. And 2 types of polymer generally used widely into the polymer based formulation of protein. Number 1, natural polymers, another is the synthetic polymers. So better natural polymers or sometimes synthetic polymer used, but it must be non-toxic, biodegradable, biocompatible, non-reactive and non-sensitive to the drugs, or even this polymer do not interact with the protein molecules. Natural polymers like collagen, obviously it will be present in our body, so there is no problem. Hemoglobin and gelatin, all are the very natural polymers. These are used for the carrier for the drug molecules. Synthetic polymer mainly the polyester like PLA or PGA. These are the 2 types of synthetic polymers, polylacrylic acid and polyglutamic acid, which are the 2 polymers that will be held for the formulation of proteins. Diffusion of the drug out of the polymers. Suppose these are the polymer and the drug is incorporated inside the polymer, it will be diffused out. Otherwise it is a problem, and the drug released by the polymer degradation. If the polymer is degraded, then the drug will be released. This is the common concept or common process by which drug must be diffused and drug must be released after degradation, and polymer will be released. Next, the liposome based drug delivery system. Liposome, it is a spherical vesicle with a phospholipid bilayer. They are the phospholipid bilayer, and in this vesicle contains some drugs or proteins. Protein which are administered through orally, it will be degraded, or parenterally there is no degrader. But if you polymer, I think a liposome based drug delivery is the most successful drug delivery for the protein drug delivery. And liposomes are the microscopic vesicles and composed of 1 or more aqueous compartment. And liposomes are the protein drug delivery. For example, lecithin used to control the drug release. Lecithin, it is a lipid containing substances which is layered the or cover up the protein molecules. That means they are not attacked by the water or hydrolysis of proteins. So lecithin is a lipophilic substance which layer up or cover up the protein molecules and to release the control release drug dosage form. Liposomes in the peptide drug delivery like bleomycin. Bleomycin, a peptide with anti-tumor activity. That means anti-cancer drug bleomycin, which is formulated as a liposomal drug delivery to reduce the normal tissue toxicity. Because direct, if you administer the bleomycin, this bleomycin normal tissue can be hampered. So if you used the polymer based or targeted drug delivery system, it is only targeted in the cancer cell. The cancer cell will be destroyed after the polymer or liposome will be removed. And negatively charged liposome produce a prolonged hypoglycemic effect in the diabetic drug, and which is injected by the negatively charged liposome produce a prolonged hypoglycemic. In case of hypoglycemic drug or insulin protein which is form a liposomal drug delivery, there is a problem of in case of diabetic drug which is injected by, that's why it is injected by the subcutaneous route. What are the advantages of the liposomal drug delivery system? Soluble in both the organic and the aqueous media. Aqueous and the organic solvent. I think this is the most important area because solubility is one of the most important factors to absorption of the drug. If you absorb, there is a distribution, then metabolism, then excretion, so therapeutic viability. Ultimately, the viability will be increases if the absorption is higher. And liposomal are the important for the targeted drug delivery directly into the liver, brain. That means the targeted drug delivery can be possible in case of you prepare a liposomal drug delivery system. And liposomes easily cross the blood-brain barrier. Liposomes easily cross the blood-brain barrier. So in case of meningitis and other types of brain disease, in that case, liposomal drug delivery you can apply it. Say for dopamine converted into the L-dopa. Used as a vehicle for the vaccine also. And the disadvantage also, it is a less stable and easily susceptible to oxidation because, and hence liposome are the replaced by the noisome alter of the liposome. Another is just like a liposomal drug delivery systems. Another is a hydrogel based drug delivery system. What is a hydrogel? Hydrogel are the three-dimensional network of the hydrophilic polymer that are insoluble. That are insoluble, but protein are soluble in nature. The protein is covered up with a hydrogel. This hydrogel cover the protein and which is released in the targeted area. This hydrogel are the polymer which have ability to soil into the water, and biodegradable hydrogel are used due to the biocompatibility. Obviously, this hydrogel must be biocompatible as well as biodegradable. Otherwise, the drug will not be released from the core material. For example, hydroxy methyl acrylate, which are used to minimize the mechanical irritation of the surrounding tissue. Example is the very common hydrogel example. And actually, in this formulation, few preparation is available in the, and go through the internet to get the more number of examples. Hydroxymethyl acrylate. I mean, hydroxymethyl acrylate which are used in the form of hydrogel preparation. Next, emulsion-based drug delivery system. This is the very common, you know it, the emulsion-based drug delivery system, because the protein is the hydrophilic nature, it is destroyed by in case of aqueous, but this protein with the emulsion, oil base, that will be not degraded in presence of water, but at the target area it will be degraded. The emulsion can be used to parenteral drug delivery for protein and the peptides used to prolong the release of the drugs. As for example, subcutaneous administration of muramyl dipeptide in water in oil emulsion. It is used for the potentiate the immunosystem. তার মানে মিউরামাইল ডাইপেপটাইড, বি কেয়ারফুল, বি রিমেম্বার ইট or write down this muramyl dipeptide WBO emulsion which is protect the or potentiate the immune system. But if you direct use muramyl disulfide without emulsion, that will be muramyl dipeptide will be destroyed. So emulsion, very simple formulation, this muramyl disulfide which is acts as a immuno booster or immune system increases. Cellular carrier protein peptide can be incorporated in the erythrocytes to achieve the prolonged release of the or targeting. Erythrocyte just like a carrier because it will be carried throughout the body. An erythrocyte used a carrier, that means the molecules, the protein molecules which is targeted to erythrocyte and bind to the erythrocyte, and erythrocyte circulate throughout the body and finally release the drugs. And release of erythrocyte as a delivery systems for the C-reactive protein and mainly used for the targeted drug delivery system and spleen, liver and spleen. Target in the liver, spleen, suppose in case of liver cancer or in case of splenomegaly or in case of the spleen infection, in that case ওই যে সি রিঅ্যাক্টিভ আগের দিন বলেছিলাম, the C reactive protein which is very highly in case of the corona or any other types, বিশেষ করে in case of cancer also the C reactive protein is the high. So in that case the targeted drug delivery system can be done by the cellular carrier system or erythrocyte used as a carrier materials for the liver and spleen. Next is the pump. Next for the list, the type of pumps. One is the implantable pumps. Drug is implanted subcutaneously and delivered by the IV infusion. Pump are filled with a drug solution and septum with a needle, and pump is delivered to the central vein 70 to 40 days at a constant rate. So in case of pump delivery system of protein, there is some several advantage. Drug is subcutaneous delivered to IV infusion, or pump is a filter through the septum with the needles, and pumps deliver drugs to the central vein of 7 to 14 days at constant rate. So long term, in case of long term therapy, this pump is very useful. Another is a mechanical pump, which is implantable pump. Another is implantable by implantation. Another is a mechanical pump, easily manipulated to the drug delivery for protein and peptides. Insulin has been successfully delivered by the portable syringe. So these are the implantable pumps and these are the mechanical pumps. Auto implements cannula, and that will be system and water high water housings. This picture is not adhesive. Device maker, angle infusion, and electrophoretic cultural program and power supply by the batteries. These implantable which are implanted under the skin, and the drug will be released, and the 7 to 14 days the drug action will be remain. In case of mechanical pump, there is a less non-parenteral route of administration. Parenteral route of is not a properly achieved. When the parental properly achieved, hence the other route are preferred. If parenteral, the most fast preferable choice is parenteral route. Another route is oral route, rectal route, nasal route, pulmonary route, buccal route, and transdermal route and ocular route. These are the 7 non-parenteral routes. Okay. Hello.
Yes, sir.
Are you feel boring?
No sir, no sir.
Just আর 60 আর মানে এই স্লাইডটা করে দিলে তাহলে এই চ্যাপ্টার মানে পরীক্ষা তো তোমার এসেই গেছে 28 তারিখ এক্সাম, তাই না? ফলে এই টপিকটা আমি কভার করে দেই, এটাই ভালো করে পড়বে। অলরেডি ইট উইল বি আপলোডেড ইন ইয়ার কি বলে ক্লাসরুম। তাহলে আজ 16টা স্লাইড আছে, আমি পড়ে দিচ্ছি, ছোট ছোট করে আছে। ওকে,
ঠিক আছে,
কারণ টাইম তোমাদের এখন অনেক আছে।
হ্যাঁ হ্যাঁ
ঠিক আছে, আর আধ ঘন্টা হলে হয়ে যাবে। ঠিক আছে,
ঠিক আছে,
কন্টিনিউ।
হ্যাঁ হ্যাঁ স্যার।
ঠিক আছে, থ্যাংক ইউ। 1 মিনিট। হ্যালো হ্যালো, কি কিছু তো আমার সাথে কথা হয়নি। আচ্ছা ঠিক আছে, আমি তোমাকে আধ ঘন্টা পরে কথা বলছি। বলি আমি এখন ক্লাসে আছি, ইনফর্মাল ক্লাস চলছে। হ্যাঁ ঠিক আছে ঠিক আছে। If you are fail in the parental route, but or it is impossible to formulate it in the parental preparation, then you either choose for the other route of administration rather than parental preparation. Number 1, oral route. The encapsulated peptide, obviously direct peptide cannot be administered orally. This is the main problem. That's why encapsulation, মানে micro encapsulation or encapsulated peptide or protein or amino acid with microsphere. Do you know the microsphere? Actually এই জন্য biotechnology বলেছিলাম, biotechnology is nothing but a single subject. It is a combination of biochemistry, microbiology, pharmaceutics and the pharmacology as well as the bio combine to form a biotechnology. The microsphere, আমি কিন্তু microsphere তোমাকে ডিটেইলস বলছি না, but you read the what is microsphere and how it will be prepared, so many process, hot homogeneous, hot compress, so there different process, but you know the what is microsphere, তার মানে in case of protein drug delivery, in that case orally preparation it will be possible that microsphere is the only one technique you can apply it for the oral route of administration. Approximately 10 micron in diameter, 10 micron diameter, সেই জন্য বলছি তো microsphere. That means the spherical size which is size less than 10 micron, and for oral used for the oral delivery system, insulin and heparin, but still now not marketed into the oral drug delivery for insulin, but it's tried to formulate the oral insulin, but still now not launched in the market. Try করা হচ্ছে যেগুলোর উপরে on research going on this insulin and heparin. ঠিক আছে, still now heparin and insulin which are administered through parenteral route, but these are under research. Orally administered insulin produce the hypoglycemic effect, heparin which is used as a anticoagulant effect, and disadvantage the acid catalyze the degradation in the stomach and proteolysis in the GI tract. Transdermal route of administration, these are the another transdermal, all of you know. This is the topical medication just under the skin or above the skin. Drug is absorbed through the skin, like example like the insulin, vasopressin. Try it, but still now it is not established. Advantage, controlled administration of the drug is possible, but theoretically, not practically এখনো হয়নি. Improve the patient compliance, obviously. যদি কেউ এমনি গায়ে ছোঁয়ালে অয়েন্টমেন্ট লাগাতে হয় তাহলে অনেক বেশি বেটার, খাওয়ার থেকে আরো সহজ। Drugs with the short half-lives and the administer, but there is some disadvantage. High intra or inter-patient variability, low penetration because of high molecular weight. If the substance is the high molecular weight, their penetration power is the low. Hydrophilicity and lipophilicity of the stratum corneum. Stratum corneum, stratum lucida. So actually there is some horny layer of skin. So hydrophilicity and lipophilicity of stratum corneum will affect the transdermal drug delivery system of different types of protein formulation. And number of approaches available in the effective protein and the peptide drug delivery. There, number 1, iontophoresis. Just you can remember, go to the iontophoresis, the sound note of iontophoresis, phonophoresis, penetration enhancer, and prodrug. Prodrug, which are the drug applied in the original form. This original form not a therapeutic effect, but after metabolized in the liver or any other organ, they are a active form of the drugs. What is iontophoresis? Used for the local and systemic delivery for protein and peptides. And in this electric current, ion means electric current is used to drive the molecular across the skin surface. Example, transport of insulin using iontophoresis. তার মানে ইনসুলিন কতভাবে প্রিপারেশন করে, একটা তো এস্টাবলিশ রয়েছে সাবকিউটেনিয়াসে, এছাড়া ওরালি অর ট্রান্সডার্মাল প্রিপারেশন করার চেষ্টা করছে, বাট স্টিল নাও নট অ্যাভেলেবল ইন দ্য মার্কেট। Phonophoresis, it is absorption in the enhanced by the thermal effect, that is the ionic effect, it is the thermal effect of ultrasonic waves and subsequently alter the physical structure of the skin surface. So these are called the phonophoresis. And the pulmonary route of drug administration, যেগুলো nebulizer হিসেবে ইউজ করা হয় থাকে, lungs are the attractive site of the systemic delivery for the proteins and peptide because of their enormous surface area around 70 square meter. Alveolar lungs are the absorption site and drug are absorbed through the lungs by simple diffusion, carrier mediated transport system. Either simple diffusion, there may higher concentration to lower concentration, or by the some carrier used for the drug diffusion. What is the advantage? Decrease in the dose requirement, fast metabolism, and increase in the patient compliance. Disadvantage, inflammation may be observed in the lungs and degree of viability was less due to the hydrolytic enzymes present in the lungs. Rectal route of administration, this is another method you can apply it, because rectum is the highly vascularized body cavity and their surface area also very high. The rectal mucosa is divided to villi and the drug are in the form of suppository gel and powder form like insulin and calcium. তার মানে ইনসুলিন কত রকম ভাবে, so many type, there is so many way try to administer. An advantage, reduce the proteolytic, because in that area there is no proteolytic enzymes, there is no chance of proteolysis. Improve systematic bioavailability with a co-administration enhancer like surfactant. In case of rectal preparation, protein formulation, protein with surfactant, there increase the bioavailability. Large dose can be administered because it is the larger the surface area. So the excess amount of drug can be administered by this rectal route of administration of proteins. Ocular, this is the smallest area, but in this route, encaphalin, thrombopoietin, releasing hormone, luteinizing hormone, glucagon, insulin are administered. Small amount of encaphalin which are used as a analgesic compound, erythropoietin, erythrotropium releasing hormone, luteinizing hormone, these are the hormone. And buccal route, the mucoadhesive dosage form can be used for the buccal route. An adsorption enhancer like salicylate or surfactant is used to the protein and peptide delivery through the buccal route. Example, এদিকে এটাকে কি এখানে তুলে দিয়ে চলে গেছে। Buccal route or mucoadhesive. Mucoadhesive. In this route, you can use, for example, oxidation, vasopressin, insulin are reported to absorb through the buccal mucosa. An adhesive gel, patch, and tablet can be used, but all are in the research level, not available in the market still now. Insulin is absorbed through the buccal mucosa in presence of the sodium glycolate. The drug are absorbed through the oral mucosa mainly through the non keratinized region. Advantage, it can be attached or removed without or any discomfort or the pain. Well, acceptability of the patient, because all of you, oral route is much more preferred than the parenteral preparation. And well acceptability, that's why the patient acceptability is much more high. And the drugs are absorbed very rapidly. And same as the disadvantage of the buccal mucosal route, administration time is limited and drug loss by the accidentally swallowing. Because buccal route, if you swallow it, then there is a less amount of drug in the present in the buccal cavity. So just like a oral route of administration. So in case of buccal mucosa, there is a less drug will be absorbed. Next is the nasal route of administration. Another route of administration is the nasal route, by which you can use the protein drug delivery systems. And the nasal absorption through the passive diffusion. In case of nasal, just like a spray insulin, I mean, that means you can see it, the insulin still now it is available in the parenteral preparation. Now there's so many methods, oral, rectal, buccal, nasal route are used for the preparation of insulin and human growth hormone also. Advantage, rapid onset of action in case of nasal. That's why sometimes spray, some drug addicted, they also just the nasal inhalers. And in case of sometimes steroid, which are used by the nasal route, because the rapid onset of action, and also simultaneously to avoid the fast pass metabolism and better drug absorption. And same disadvantage also, in case of long term uses, causes the toxicity in case of nasal. And size of protein and peptides drug reduce the systemic viability. If the smaller the size, there is a increase the bioavailability, but if the larger the particle size of the protein, then the bioavailability will be reduced, because nasal having some porosity. So the higher molecular substance cannot be administered by this nasal route. The protein formulation, protein sequence, the modification site directed by the mutagenesis, PEG glycation, protein annihilation, microsphere encapsulation, and formulation with the permeabilizer. Glycation, PEG glycoacylation. Number 8 is a non-toxic, hydrophilic, and FDA approved Food and Drug Administration approved unchanged polymer. PEG glycation, next time I thoroughly discuss about this type of formulation used in the, and increase in the in vivo half-life also, and decrease the immunogenicity. That means the immunogenic activity will be decreased, and increase in the protease resistance. That means protease which is mainly short করে লেগে যাবে কারেন্ট, ওইটা সরাও না। Increase the protease resistance. If the protease resistance will be increased, there is a degradation or proteolysis will be prevented. Increase in the stability also. In case of glycation, there is a stability problem or higher the stability than the any other dosage form. And protein annihilation, attachment of the addition or the secondary or non-immunogenic protein for the in vivo protection, and cross-linking with the serum albumin. Increase in vivo half-life also, and cross-linking and connecting the protein engineering with the antibody fragments. Suppose these protein drugs with the SSC antibody, sac fragment proteins, and finally this antibody bind with the protein drugs, and they are used for the formulation of the protein annihilation. The formulation of the with the permeabilizers like salicylate, aspirin, fatty acid, omega-3 fatty acid, and the metal chelators, EDTA. Marketed formulation as a protein, given some few examples. Lupron as a stabilizer, lyophilizer microsphere contain the leuprolytic acetate, route IM intramuscular, indication for use or indication for prostate cancer. And pitracin tannate, vasopressin tannate in the peanut oil, peanut oil. All of you know this peanut oil intramuscular, it is a anti-diuretic effect. On HP actor gel, adrenocorticotropic hormones in the gelatin, intramuscular and subcutaneous endocrine cancer. Sandostatin eye depot, octreotide, intramuscular, here mentioned here, neutrophin, the growth hormone in the IM for drowsiness. So these are the formula marketed product used for the protein formulation. Another is the given some the formulation metrodin, proganin, profas, esper, glucagon, the glucagon IMSC and IV, hyperglycemia, leukemia, infertility, progels for the infertility or induction of the evolution and the hormone deficiency. Obviously, you prepare some formulation, their stability problem will become. So how to minimize the stability? This capability of the particular formulation in the specific containers and the closures system to remain within the physical, chemical, microbiological, toxicological and the protective specification. Obviously, the stability most important factor. Suppose you prepared a formulation, good formulation for any route of administration, but after some times or after few days or few months, it will be degraded. So be careful, be serious or be attention for the stability problems. So the stability, at least 2 years must be stable. There will be problem in case of purchase or in case of commercial aspects. The evolution of effect of the environmental factors on the quality of the drugs and substances, the formulated product which is be utilized for the prediction of the shelf life, determines the proper storage condition. This is also another most important factor because the protein after before the storage condition in the too high temperature or low temperature or the pH changes or some metal ion or some presence of oxygen. So there is some stability will be problems. So in case of general selection of the batch, container or closure system, specification, testing frequency, storage condition, stability contaminator, evaluation, statement, labeling and ongoing stability study. These are the effect you can study. After that it will be formulated or launched in the market. So protein and peptide based pharmaceuticals are rapidly becoming a very good important class of therapeutic agents that will be discussed in the very earlier and are likely to replace many existing organic based pharmaceuticals in very near future. So be take care, be interested in this field. The protein used as a drug in the different formulation and which is more acceptable than the conventional dosage forms. Peptides and protein drug will be produced a large scale of biotechnological process and will become commercially available for the therapeutic use. And there is a need, their need in the clinical therapeutical regions and intensified the investigation for the convenient and efficient delivery through the non-massive system. So nowadays, in future, this protein drug delivery system is more increase and increase. So if you interested them for this formulation, this type of just pharmaceutics people which are interested in the pharmaceutical preparation, conventional dosage form, but you are prepared same drugs or protein drugs by this different type of formulation, and this is the most growing type of formulation. So be interested. If you understand, please like me, or if you not understand, please comment me in my YouTube channel or in the Google Classroom. These are the reference books, control drug delivery concept. And then the VAS and the understanding and foundation of the peptides, protein and peptides. And bioprocess journal trends to involve this process, and these are the, you can see it, look it for the better understanding. Thank you very much. If you understand,
ওকে স্যার, ক্লিয়ার স্যার, বুঝতে পারা গেছে।
হ্যাঁ,
হ্যাঁ স্যার, বুঝতে পারা গেছে।
বুঝতে পারা গেছে?
হ্যাঁ স্যার।
জাস্ট এতদিন তোমরা সিউটিক্স, ইউ ক্যান ইন কেস অফ সিউটিক্স ইউ আর প্রিপেয়ার ডিফারেন্ট টাইপস অফ formulation, but here main target is the formulation of protein by the different types of formulation. Just you understand?
ওকে স্যার।
Okay, so this material will be uploaded in the YouTube channel or in the Google Classroom. If you like, please like me, or you comment or give a feedback in this class. Just in the comment, just roll. So one topic is covered, protein drug delivery system or different types of drug delivery systems. Just make a comment, a few word or sentence. Make a comment what do you understand, or based on the understanding, based on the knowledge, give a feedback in the instantly. এই চ্যাপ্টারের উপরেই পরীক্ষা হবে কিন্তু এখন, ঠিক আছে? ওকে
ওকে স্যার।
your syllabus for this chapter, for be careful this, you can just this follow this ppt and carefully read it carefully. I think it will be all the ppt uploaded already. ঠিক আছে, just to comment on the chat box.
স্যার বলছিলাম স্যার 28 তারিখে যে রুটিনটা কবে দেবেন?
রুটিনটা তো এখন বেরোয়নি, হয়তো আজকে বেরিয়ে যাবে, বেরোলেই আমরা গ্রুপে দিয়ে দেবো।
ঠিক আছে,
ওটা তো ওটা পারিয়াসের বা প্রিন্সিপাল ওরা দেখছে, ওটা তো আমাদের কাছে নেই, কিন্তু পরীক্ষাটা হবে হয়তো। ওটা কবে এবার কোনটা আগে কোনটা পরে সেটা আমি বুঝতে পারছি না, ওরা বেরোলেই বোঝা যাবে।
স্যার আজকে তো 26 তারিখ হয়ে গেল না, সেই জন্য।
সে তো আমি বুঝছি, কিন্তু যারা দেবে তাদেরই তো আগে মানে হিসাব মতো কালকেই ওটা বের করে দেওয়া উচিত ছিল। এখন আমি বললে তো আর হবে না, তারা ভাবছে তাদেরটাই কি কারণে আটকাচ্ছে আমি সেটাও জানি না। কিন্তু আমি হলে তো, যেমন আমার যে পরীক্ষা হবে 5 তারিখ, আমি 1 মাস আগে নোটিশ বার করে রুটিন দিয়ে দিই। মানে
এটাও হতে পারে যে অন্য কোনো মানে বি ফার্মের কোনো এদের পরীক্ষার মানে রুটিন করছে হয়তো স্যারেরা, এটাও হতে পারে, সেই জন্য প্রবলেম হচ্ছে।
ঠিক আছে, বি ফার্মের রুটিন করছে, তোমাদেরও তো একই টাইম দেওয়া হয়েছে, তোমাদের তো সহজে বের করে দিচ্ছে, যাদেরটা দেওয়া যায় সহজে। আমি কাজটা যেমন আমি যেমন একা করি, এটা সহজে দেওয়া যায় সেটা কিন্তু আমি সহজেই সেটা আগে বের করে দিই। কিছু লোক তো পেলো,